Ruby Madam for the presentation. And on behalf of Mahatma Gandhi University, we take this opportunity to express our sincere thanks for your kind support. And this is part of some seminar series which are being conducted as part of ANRF Pair project. And we are also interested to have much more collaboration with the Center of Excellence in Nutraceuticals for is specifically related to the diabetes and fatty liver. That is a major two conditions that we are into. And we would be we are very much happy to have your feedback on how some collaborations can be initiated for this type of project. And for the basic fact of the project is that it is a project that is funded by government of India in a hub and spoke model, where the hub institution is University of Hyderabad, and there are six spoke institutions are there, and one of the spoke institution is Mahatma Gandhi University, and the total cost of the project is itself is 120 crore. So it's a mega project, 5-year project, and we will be much more happy to have your all the support from Madam. And to the participants, Madam is very highly supportive, dedicated scientist, and has spent years at Rajiv Gandhi Center for Biotechnology in various aspects of cancer biology. And during the work, Madam was much more helpful as a mentor. And actually, I completed one part of my PhD thesis under the guidance of Ruby Madam. And we have Professor Smita is also here, and we were there at RGCB. And Professor Smita is a student of Professor Ruby Madam. So it is just like a connecting with our mentors and connecting with our well-wishers who supported us always. And with these words, I now invite Chandana to introduce Madam to the participants. Thank you very much, Madam.
Thank you. Thank you, sir, for the warm words. So today, we are moving ahead with our first webinar series under the ANRF Pair project. And good afternoon once again, and welcome all the respected dignitaries and faculty members, research scholars, and dear friends. So today we are really blessed to have with us Dr. Ruby John Anto, an internationally renowned scientist in the field of cancer biology, nutraceuticals, and translational medicine. Dr. Ruby John Anto took her PhD from Amala Cancer Research Center, Thrissur, started her career as research associate and later as scientist C at RGCB, Thiruvananthapuram, and retired as scientist D in 2023. She has also served as a visiting scientist at MD Anderson Cancer Center, Houston, USA. Currently, Madam is serving as the Chief Scientist at the Center of Excellence in Nutraceuticals under KSCSTE, Government of Kerala, and also as Honorary Scientist at the Institute of Advanced Virology, Thiruvananthapuram. She is the recipient of several prestigious national and international recognitions, including the National Women Bioscientist Award from the Department of Biotechnology, Government of India. Dr. Ruby Anto is a Fellow of National Academy of Sciences and Kerala Academy of Sciences. Madam holds national and international patents, awarded 14 PhDs under her supervision, and trained 15 postdoc fellows. She has an H index of 42 and more than 7,500 citations from around 82 international research publications in high impact factor journals. Her research focuses on bioprospecting for natural products having therapeutic potential against cancer, inflammatory disorders, and other lifestyle diseases. Her pioneering research led to the discovery of Uttroside B, a promising anti-liver cancer molecule that received US FDA orphan drug designation and multiple international patents. We are indeed privileged to have Madam with us today. I request all the participants to actively interact with Madam during the session and make the best use of this opportunity. Without further delay, I respectfully invite Dr. Ruby John Anto to deliver today's webinar session titled The Therapeutic Promise of Solanum, Nigrum, and Uttroside B. Thank you all, and wishing everyone an insightful session. Over to you, ma'am. Thank you.
So thank you so much for the wonderful introduction. And definitely it is the most proud moment for any scientist to see the students whom she has mentored is in covetable positions in prestigious institutes. And I'm so happy that Dr. Radhakrishnan and Dr. Smita is there as professors. I'm really happy to present the work. And I'm very happy that I have done something good because I am continuing my research through my students. So I'm so happy, and I thank you very much for inviting me to present my work in this webinar. So I'll start my presentation. So today I'm going to present the work we have done on the plant, Solanum nigrum limb. This is a medicinal plant, and we isolated a compound Uttroside B from this plant in our lab, and its efficacy as a against fatty liver disease and its conversion to liver cancer. So NASH or MASH, currently this NASH, non-alcoholic steatohepatitis, being called as metabolic dysfunction associated steatohepatitis. So this is this can sometimes lead to cirrhosis, or it can progress to HCC also. So it is characterized by the excessive accumulation of fat in the liver. And in the global epidemiology, the most important affected places are UK, India, Germany, and the Middle East. So who is at risk? It is established risk factors are obesity, type 2 diabetes, dyslipidemia, hypertension, and insulin resistance. So the current management is we don't have much medicines to manage this NASH or MASH. Currently, it is being managed with vitamin E, thiazolidine diones, and through lifestyle modification. And the only FDA approved drug for the treatment of MASH is resmetirom. So coming to our work, this Solanum nigrum limb, it is commonly known as black nightshade. It is widely used in traditional medicine against liver ailments, and it has many other properties such as anti-inflammatory, antioxidant, anti-tumor, hepatoprotective, and immunomodulatory potential, according to the Ayurveda literature. And we isolated from Uttroside B from the methanolic extract of Solanum nigrum leaves. And it has a very high cytotoxic potential towards liver cancer cells. Actually, we compared the cytotoxic potential of this Uttroside B with sorafenib, the first-line treatment option for liver cancer, the FDA-approved drug. So we found that Uttroside B IC50 was 500 nanomolar, while that of sorafenib was 5.8 micromolar. And we compared the activity in vivo, but we did not get a tenfold activity for Uttroside B in the in vivo. But still, we found that Uttroside B is more potent than sorafenib. And we also found that it doesn't have any toxicity, while sorafenib treated animals showed some toxicity, such as the blood stains in the fecal matters, etc. So actually, the isolation was done by Dr. Lakshmi R. Nath, who did PhD under my guidance. And currently, she is a professor at Amrita. And the continuation was done by Dr. Shweta. She is doing postdoctoral studies at US. So this efficacy, this anti-cancer potential of this compound was patented in different countries, USA, Canada, South Korea, Japan, and Europe. And the technology has been transferred to the company QBiomed, a US-based company. And this currently this drug is a FDA approved orphan drug against liver cancer. So actually, this is a very potent molecule, IC50 500 nanomolar, but the size of the molecule is very big and it is very difficult to synthesize it. Actually, for the companies need a synthesized form of the compound. It could be synthesized, they succeeded in synthesizing, but the cost was too high than for the isolation isolation of Uttroside B from the plant itself. Just like our paclitaxel. Paclitaxel can be synthesized, but still it is being isolated from the plant, from the bark of the specific nutrient. So this is the condition for Uttroside B also. So, since the technology is already transferred, we cannot take the Uttroside B as a drug anymore. But what I did is since this Uttroside B is a water-soluble molecule, so we compared whether this Solanum nigrum extract containing the IC50 dose of Uttroside B will give the same activity. Of course, I don't claim that the efficacy of the extract will be only because of Uttroside B. Definitely, there are more compounds, very potent molecules itself, but their IC50 is not in nanomolar levels, in micromolar levels, around 25 micromolar. So we compared the efficacy of Solanum nigrum extract, Uttroside B and Solanum nigrum extract containing IC50 dose of Uttroside B. We got almost the same effect for both the groups. So that means it can act as a substitute for the for Uttroside B for the time being. So we okay, and we already saw that only one molecule is one drug is the FDA approved medicine for managing the MASH. So many people with fatty liver disease and liver cancer approached us, and actually it was due to their compulsion we started the clinical trial. Actually, it was not a clinical trial; it was like a toxicity evaluation. That is how we got the clearance, ethical clearance. But there are many people who joined in the study. There were liver cancer patients, there were fatty liver disease patients, there were cirrhosis patients, and we also had normal volunteers. So this is the study. It is still going on. So there is we know that we have many saponins will induce the hemolysis. So that is what we first checked whether this compound, it is a saponin, Uttroside B is a saponin, phytosaponin. So we checked whether it will induce hemolysis using the blood collected from healthy volunteers. We found that it doesn't, neither Uttroside B nor sorafenib induce any hemolysis. So before starting the clinical trial, giving into the patients, first we did studied in some 16 healthy participants, whether it induces. Actually, many people, when they present, they say, I heard one presentation in our science congress, one very from a scientist from a very reputed institute saying that there is a scientist in Trivandrum, she is giving Solanum nigrum extract to people, it will induce kidney adichu povum. That was the language he used. So for that is what we did first. Actually, this was we cannot start a trial without doing a normal study on healthy volunteers. You know this is, I am the first person did this thing. I am the so there are many people who participated in this one. So we did not find any change for the urea, creatine, uric acid, no change. Okay, so this we did for more than 3 months. Then only we started giving this to the these are all other parameters. This is a violin plot for the same study. This is liver function, all the liver function enzymes, then bilirubin. So we checked everything. Okay, for the lipid profile, we found there is a reduction in the lipid profile level also. We did not find to any of the participants there was a hike in any of the parameters. It was or in all the 16 participants. So this is a study currently going on. So now there are 72 participants of median age 54. And there are 26 women and 46 men, and healthy subjects are 16, fatty liver patients 24, chronic liver disease MASH patients 11, and liver cancer patients 21. Okay. So now coming to the results of that study, so we found that there is we did not find any change in the kidney function. No, there was no hike in the kidney function of any of the patients. So this is we can see there is a reduction in the cholesterol, triglyceride. There was a significant reduction for the cholesterol, triglyceride, HDL, LDL, everything. Okay, HDL, there was we did not find a change. And LDL, we found a reduction. This is liver function. This is the alkaline, this liver function by Violin plot. This is urea, uric acid, creatinine. We did not find a change. So this is bilirubin. So this is the comparison. So these are the patients. Yeah, around 7 patients. We can see the reduction. Okay, from the range is 37. So from the first month, when in the starting it was 63, then it came to 21, 14. So that means it is less than. 37 is the normal range, so it comes down. And SGOT also 45 to 15 and 60. So for all the patients, we are seeing a regular reduction in the values. So this is a patient from actually he was a security in Rajiv Gandhi Center for Biotechnology. And he was diagnosed of chronic liver disease on 2019, and he was told that he will die in 2 years. That's what the doctor told him. So he came to our lab and asked whether I can have this. So in 2020, he joined the clinical trial. So this is his report. In 2020, it became grade 2 fatty liver. Again, after 6 months, it became grade 1 fatty liver. Even now, he is not dead. He is a security still at RBCP. So, and in liver cancer patients, of course, it is not a bundle drug. If a person with very high serum profile and HCC, IFP level and all, we did not get. In some patients, in the last moment, somebody joins. So definitely, it doesn't make any progress. But in other cases, we are seeing a reduction in the AFP levels and PIVK. And we checked whether this Uttroside B is present in the Solanum nigrum extract. This is Uttroside BP. So this is the MS spectrum of aqueous extract of Solanum nigrum. So definitely my lab was a cancer biology lab. All the previous results were of the liver cancer study. I showed the first 2 papers. So we are seeing very good results for this fatty liver disease also. So we thought, okay, we cannot say that just Solanum nigrum leaf is just because it is showing very good efficacy, the result is due to only Uttroside B. So we checked whether Uttroside B can be used as a therapeutic option, just like resveratrol can be used as a clinical remedy for the MASH. So we start as the first. This is the in vitro study. We can say that. So this is the palmitic acid. So this is with the Uttroside B. We can see Uttroside B reduces the accumulation of lipid droplets. Accumulation of lipid droplet is an important stage for the fatty liver disease. So this is G2 cells treated with the palmitic acids. So there was a significant reduction. So, and we checked other molecules, and actually in our one of our studies, that is using cancer cells, we found that it induces the blockade of the Uttroside B induced autophagic cross-survival signals augments its chemotherapeutic potential. That means this compound induces autophagy. This is a molecule which induces autophagy. So we checked whether in this palmitic acid, this Uttroside, whether Uttroside B can reduce the induce the AMPK value. So we found that it is inducing. So this is FAS and ligand. So Uttroside B phosphorylates AMPK and reduces the expression and downregulates the FASN. So then we checked its therapeutic efficacy in vivo conditions. This we used two models. The first model was high fat diet induced fatty liver model. So this is the model, and these are the representative images of the livers of mice fed with HFD, displaying characteristic features of MASH, hepatocyte ballooning, and steatohepatitis. Okay, all the pathological examinations and immunological studies were done by Dr. Shankar Sundaram, verified by. So we can see the therapeutic efficacy of Uttroside B in vivo. We can see these are the livers of the control animals. It looks really bloody. But when in the case of HFD mice, you can see the pale liver. And again, after when we treat it with Uttroside B, we get the earlier structure. Okay, so this is the HFD group. This is the HFD plus Uttroside B group. So these are the serum biochemical parameters. We checked all the parameters. We did these are the values we could find at down in the HFD date. We can see an induction of triglyceride, all the liver parameters, while it is reduced due to in the HFD plus Uttroside B group. Then we did another. Actually, this is another. These are the different staining methods to see whether it is the change of molecular changes are happening. So we did the hematoxylin using staining, oil redo staining, and serious red staining. So we can see in the high fat diet group, the steatosis macro. Yeah, this is the high fat diet group. Here we can see the steatosis macro and microvesicular hepatocellular ballooning, you can see here, and lobular necroinflammation and fibrosis, while in the case of Uttroside B treated group, it was normal histology with the mild steatosis. These are all diagnoses by Dr. Shankar Sundaram. He was the HOD of the Department of Pathology, Government Medical College, Kottayam. And histopathological analysis, the HFD mice displayed characteristic features of MASH, such as formation of mammillary dung bodies, lobular necroinflammation, and hepatocellular ballooning, whereas Uttroside B treated mice liver displayed a normal histology without any of the characteristic features of MASH. This is the Uttroside B treated group. This is oil redo staining. This is oil redo is a fat soluble hydrophobic lysochrome dye used to selectively visualize neutral lipids and triglycerides in tissue cells. So you can see the staining in the HFD group, while it is significantly reduced in the Uttroside B treatment. So the Uttroside B treatment reduces the deposition of lipid droplets within hepatocytes. And inflammatory markers. There are many inflammatory markers such as MCP1, TNF-alpha, interferon, and CRP. All these levels are significantly reduced by Uttroside B compared to the NASH group. And the liver tissues of MASH-induced mice treated with Uttroside B exhibited decreased fibrotic connective tissue and downregulation of mRNA expression of collagen alpha. Okay, so serious red, it is being an anionic dye, selectively binds to the serious red, will bind to the basic amino acids of collagen molecules, and you can see the serious red staining in the HFD group. And trichrome staining, this is a much more reliable experiment. So here the nuclear staining, Weger's hematoxylin stains cell nuclei black, and the initial cytoplasmic acidic staining, a mixture of this scarlet and acid fusion stains cytoplasm, muscle, and collagen red. Okay, you can see very clearly in this HFD group, the staining, trichrome staining, and which is completely reduced in the case of Uttroside B treatment. And this is activation of these HSCs lead to accumulation of collagen, and the marker for hepatic cell, that is HSCs, hepatic stellate cell activation. This is an important marker. Alpha SMA activation is an important marker for fatty liver disease. So we can see that Uttroside B completely, almost completely, removes the alpha SMA activation. Yeah, this is in the tissue samples, the staining. We can see this is control. This is alpha HFD group with the alpha SMA staining. This is again, it is completely reversed. So this is our previous paper, not on NASH. This is liver cancer. We found that this Uttroside B will induce the autophagy, and so we checked whether this autophagy in happening to normal cells can be blocked by the what is happening to the in the case of normal cells. Okay. Yeah. And so Uttroside B induces autophagy in HFD in the mice tissues. We can see it induces in the high-fat diet-induced mice, it induces autophagy and ameliorates the characteristic features of NASH. So these are all the HFD-fed mice. So you can see the induction of autophagy by the Uttroside B. So we can see a very significant increase in the autophagy. This is LC3. We can see here LC3 also is increased by Uttroside B. Now, this is another study. This is to see whether Uttroside B can prevent the progression of NASH to HCC. So this is a stam mice model. This is a streptozotocin plus this fatty liver, high-fat diet model. So streptozotocin is administered neonatally to produce mild systemic inflammation. So this is just like a type 2 diabetes case, depicts the pathogenesis of human-like MASH-induced HCC. So this is what so it will take from 20 weeks to 22 weeks to the onset of fibrosis happens in 9 to 12 weeks. So it is given in very small 2 days old babies to the first injection. So yeah, day 2 is the streptozotocin injection. Then this is the protocol. So in 22 weeks, it is the animals are concentrated. So we can see in actually there is a concept that all liver cancer studies should be done in males because females will not get this liver cancer. That is the concept. So in all animal models, if you see, they say that we will take only the male animals. But at the 2 days we cannot wean. We cannot know who is male and who is female. Okay. So what we can do is just take all the animals. Okay. Then we, after taking this, then we can remove the females. So we thought definitely. Anyway, actually it is not true that females will not get liver cancer and fatty liver disease, not liver cancer, fatty liver, because my mother died of fatty liver, I mean cirrhosis. So I thought definitely the women are getting fatty liver disease and cirrhosis. So let us try. Anyway, we have to kill these animals, female animals. Let us give Uttroside B treatment to the females also. So we continued the study with the males and females. Okay. So what happened is that in males, all the males got cancer, these changes. Okay. But in the case of females, at the day 20, 20 weeks, there was not many animals, but when we kept for maybe some one more week or 2 more weeks, they also developed tumor, but not all. Okay. So, and we can see there goes a reduction. So we can see here all animals got these are males. Okay, but this is tetrodotoxin. So females in some cases they did not get. So this is the histopathology analysis. So males, well-defined HCC, intranuclear intrusions in all animals. But in some animals, females, we got HCC. In some cases, we got steatosis, and some we got normal animals. That means not all animals develop. So the prevalence of getting liver cancer in females are less, but we cannot say that it will not develop. Okay. But in the case of Uttroside B treated animals, all animals showed normal histology as diagnosed by Dr. Shankar. And Uttroside B reduces the expression of tumor proliferation marker KSCSTE in MASH-induced HCC mouse model. We can see this. You can see that KSCSTE is completely downregulated by Uttroside B treatment. Then collagen accumulation. We can see this is liver, this is tumor. Okay, this is on the tumor, this is the liver. So you can see that in the case of Uttroside B treatment, this serious red and trichome. In both the cases, we can see there is a complete reduction of the lipid accumulation and the collagen. This is the alpha SMA. We can see the alpha SMA is completely downregulated in the tissues of Uttroside B animals. So Uttroside B reduces the expression of alpha smooth muscle actin in MASH-induced HCC model. Then Uttroside B treatment induces, this is another hallmark of fatty liver disease, NASH, I think. So this ACC catalyzes the rate-limiting step of de novo lipogenesis and regulates fatty acid beta oxidase in hepatocytes. So we can see, yeah, in the case of Uttroside B, there is a upregulation. So this is the injection. Okay. And Uttroside B treatment induces overexpression of acetyl CoA, carboxylates, and phosphorylates the enzyme at the RIN 78. There are reports like that. Then we did a transcriptomic profiling of STAM versus Uttroside B treated STAM mouse model using the nanostring encounter assay against a set of genes associated with the fibrosis. We found a clear reduction. So this is the Uttroside B group. This is the high-fat group. And this is the nanostring analysis of mouse fibroid, downregulation of genes involved in collagen and DCM biosynthesis, including all these molecules, were downregulated on Uttroside B treatment. This is the Uttroside B group. And Uttroside B downregulates fibrosis associated pathways, MASH-induced HCC mouse model. So you can see the downregulation of all the fibrosis associated pathways. And the comparative pathway score analysis revealed a marked downregulation of fibrosis associated pathways such as ECM synthesis, collagen biosynthesis, and myofibroblast regulation pathways. And the Uttroside B downregulates fibrosis associated pathways of MASH-induced HCC model. We can see all the PPAR gamma is very important. We can see it is upregulated. The mRNA expression of PPAR gamma, it is a very key molecule in the regulation of fibrosis. And PPARs are regarded as therapeutic targets for a variety of metabolic diseases such as NAFLD, diabetes, dyslipidemia, and cardiovascular. And Uttroside B treated samples displayed a downregulation of all these molecules, signaling coupled with the upregulation in autophagy and PPAR signaling pathway. So this is the model. So the conclusion, Uttroside B ameliorates non-alcoholic steatohepatitis and impedes the progression of MASH to hepatocellular carcinoma. So this is our conclusion, and this is a schematic representation. Regina Peterson did this work, and this work is currently accepted in published in the Journal of Hepatological Reports. So our conclusions are Uttroside B exhibit therapeutic potential against metabolic dysfunction associated with steatohepatitis. Uttroside B hinders the progression from MASH to HCC by blocking the development of fibrosis. And aqueous extract of Solanum nigrum leaf powder exhibits comparable efficacy to that of Uttroside B and can serve as a viable alternative to Uttroside B in the treatment of MASH. So the take-home message, in the current scenario of limited therapeutic options for MASH, our findings suggest that Uttroside B is a potential candidate to be evaluated in clinics against MASH and MASH-induced HCC. So thank you so much. Hello? Hello?
Hello. Hello. Thank you, ma'am. Thank you. Thank you. Thank you for the.
It was audible, right?
Yes. All good.
Everybody left.
Yeah. Ma'am, my name is Arjun Srijit. I am a project associate in the ANRF pair project.
Okay.
Actually, currently I am with your paper, Orphan Drug Uttroside B impedes MASH progression and
Okay.
HCC development.
Okay.
Ma'am, I have a doubt regarding from that study. Your study showed restoration of autophagy markers like ATG7, Basilin1, and LC3-2.
Yeah.
Yeah. Ma'am, you did investigate whether Uttroside B specifically affect mitochondrial dysfunction or mitophagy in a MASH?
Actually, this study, we did the Western blot, all this, and this RNA, all these things we did, but not specifically. Okay.
Okay.
Yeah.
Yeah. Do you think the anti-steatotic and fibrotic effects of Uttroside be partially mediated through like
Autophagy. Yeah. That's what we, because that is one of the important this autophagy is a very important, it has a very important role in fatty liver disease. And we found that it is Uttroside is strongly inducing autophagy. But in the case of liver cancer, when we block this autophagy using some chloroquine, that was our study, we have published it. When you use it, you know that chloroquine, the anti-malaria drug, okay, it is an inhibitor of autophagy. But when we mix the combination of Uttroside B with this chloroquine, it will again minimize. That means from 500 nanomolar, the IC50 further goes to 250 nanomolar. Okay, in the case of cancer, that is happening. But in the case of normal, that is different. Okay. Yeah. So it is in the case of normal liver, it is a protection. So we were thinking why this compound is such a good medicine, but why it is inducing autophagy. But in the case of cancer drug, when you block the autophagy induced by this molecule, again the efficacy of the drug increases. That means the combination of Uttroside B plus chloroquine gives the double effect.
Is it specifically affect any pathway or?
Now we think that it is AMPK pathway.
Yeah.
Then EGF signaling.
EGF.
Yeah. Actually, we did not study that in this in the scenario of this fat MASH, but in the scenario of HCC. That is another paper which is currently accepted.
Oh,
Okay. Yeah.
Okay. Yeah. Thank you. Thank you. Hello.
Hello.
Where are all they?
Hello.
Chandra madam.
Any other question? Gautam sir, do we have any question?
Students are not there.
Yes, ma'am. Ma'am, also how you feel that is during these type of studies focusing on the translating these type of phytochemical discoveries into therapeutic applications. What are the major challenges you faced? Could you please explain on that?
Yeah. Actually, it was a big challenge. Even I did this work in RGCB, Rajiv Gandhi Center for Biotechnology. Okay. Even then, nobody was supporting this study. Okay, because it was a standard for biotechnology and for molecular studies, and a plant-based study and plant-based evaluation. Even for the DBT assessment boards, I was getting negative reviews. Why you have to do? There are many other institutes for DBT for doing this. It will be difficult for you to. Of course, there was nobody else doing this type of work, so it was very tough. But could I manage it through extramural grants? And definitely RGCB definitely support for the patenting and RGCB. So otherwise we cannot do that here. Of course, RGCB gave me a very good support in the last, but it was definitely it was a fight. We have to do that. It's not that everybody. I don't think that I was walking through a flower stone pathway. It was not like that. But definitely, when you have a will, you have a way. Okay.
Okay, ma'am. So, if Arjun, any more question?
Yeah, ma'am. One more thing.
Please.
I'm exploring mitochondrial dynamics and genomics in MASLD. MASLD. So, can we do that thing in mitophagy? That Uttroside effects on mitophagy? Can we study that area?
But at present, I am not there in RGCB.
Oh.
Okay. So you have to collaborate with somebody in RGCB for that. I can also help you for that.
Oh.
Okay. If you want somebody's help also. We can also do it in a collaborative mode so that we already have an MOU with RGCB.
Okay.
So if possible, if it is a self-study and all, I can help you.
Yeah. Thank you. Thank you.
Okay, thank you very much, ma'am. If no more questions, we can wind up. I will call my fellow Miss Anna for the vote of thanks.
Thank you so much for the opportunity.
Hello, ma'am. This is Sumi.
Sumi.
Nice presentation, ma'am. Lots of data. Actually, one of the students in the project is working on trying to find a phytomedicine for fatty liver disease.
Okay.
And actually, I will be guiding him.
Okay. Good.
Okay, ma'am. So your suggestions, advice, and blessings are required. Okay, ma'am. Thank you.
Okay.
Good evening, all. On behalf of the ANRF pair team at MG University, I extend my heartfelt thanks to all those who contributed to the success of this webinar session. First, I express a sincere gratitude to our distinguished speaker, Dr. Ruby John Anto, for delivering such an insightful talk. Ma'am, your presentation provided valuable scientific insights into the therapeutic potential of Uttroside B. Thank you so much, ma'am. Next, I'd like to thank the organizers, Dr. Jayachandran K., Project Investigator, ANRF PAIR, and Dr. Radhakrishnan E.K., Principal Investigator, ANRF PAIR, MG University, for their constant guidance, coordination, and efforts in successfully conducting this webinar series under the ANRF PAIR initiative. My sincere appreciation goes to all the researchers, scholars, and the students who joined the session and actively participated through their interaction and discussions. Once again, thank you all for your valuable time and participation. We hope the session has been informative and inspiring for everyone. Thank you.
Okay, thank you, ma'am.
Thank you. Thank you.